So let's briefly overview the PCR polymerase chain reaction that has such an important role. So when we have a tiny pieces of DNA, which we want to confirm, let's say infection from coronavirus or something else, a disease, then we have to, replicate this to amplify enough amount of DNA so that we can test them correctly. So here is the original piece of DNA, original DNA, which we want to replicate. Then this is connected and we have to denature it first. For that, we need to heat that so that this DNA hydrogen bonding can be detached. And while we have this DNA primer and nucleotides around. So first, what we have to do is anneal at proper temperature, anneal the DNA primer to its proper position by hybridization. And this is the starting point for replication so that we can elongate at 72 degrees with these DNA nucleotide and polymerase. So the polymerase chain reaction happens, and on the opposite side also polymerase chain reaction. So one DNA strand become two, and we repeat this one. So it's 1, 2, 4, 8, 16. So it keeps multiplying until millions of copies can be made. So this is a very nice, cool idea, and which led to Nobel Prize in 1993. So a good example of this is nowadays, okay, if some patient or some people is suspected to get infected by coronavirus 19, how to confirm that? And in fact, this PCR helps. But coronavirus is an RNA virus, while this PCR is for amplifying DNA. So you need to make this RNA the corresponding DNA first. That requires not transcription, but the reverse. So we call it as a reverse transcription. So to check that, we need a reverse transcription PCR, because making that RNA information of coronavirus into a DNA, and then we run this PCR, and there's called a real-time PCR, which we can see in real time during these cycles to check if there is suspected DNA piece or segment is present or not. So another slide, this is for reference for PCR, a technique to produce many copies of specific regions of DNA for further analysis. So let's go back here, double strand DNA, 5' to 3' anti-parallel fashion, and we denature it at 95 degrees, and then we anneal or attach primers, 2 primers at its complementary site, and then we extend by making this DNA polymerase working to make a copy of this. The important thing to recognize here is this DNA polymerization happens at 72 degrees, which is way higher than our physiological temperature. What it means is most of our enzymes are supposed to work best at our physiological temperature, such as 37.5 degrees, but this has been the key to find. And in fact, there are in a very hot place, there's still life forms around. We found the enzymes working at this temperature, that's called Taq polymerase. And because of this finding and use, we can actually run this DNA polymerization, and you keep this cycle over and over, and we increase the copy almost exponentially. So the components for PCR, you have to have DNA template and 2 primers, 1 and 2, which is primer means short nucleotide sequences that are complementary to the ends of the target DNA. So design of primers are important, and we do need DNA polymerases, especially Taq polymerase. And using this, we can amplify or even quantify the amount of DNA, because we know how many cycles we went through, and this can be used for diagnosis of disease or microorganism, bacteria or viruses. Okay, next time we will get into more into detail of central dogma and how people have discovered the genetic code.